means the moment I receive a stimulation from the nervous system, I'm going to perform an action. So what this action could be contractility. I will contract my muscle, which means it will seemingly appear shorter and produce force, or extensibility, in which it gets longer, and elasticity. And elasticity, does anyone know what protein is responsible for muscle elasticity? And yeah, elasticity means we are avoiding overextending or tearing the muscle apart. So the functions are producing movement, obviously. Movement is not only physical movement or moving around, it's also within the body. اللي هي what? Yes, peristalsis, the movement of the intestine. Or that vasodilation, vasoconstriction of the blood vessels. Maintaining posture and position, you know we have gravity force always acting upon us, and so we need to keep our posture maintained. Stabilizing the joints because they are connected to bones and all that, and generating.
So, my approach while studying specifically biology, I use a lot of mnemonics, and I create stories in my head that makes it much easier to understand, okay? So, we'll talk about the gross anatomy. So when I say gross anatomy, I'm talking about three specific aspects. So the nervous connection to the muscles, the connective tissue around the muscles, and the attachment of the muscle to the bones, okay? So, the nerve and blood supply, I use the 1-1 plus and nav rule. What does that mean? It means every muscle will have one nerve, one artery, and one or more veins supplying it, okay? So, every muscle is supplied by one nerve, one artery, and one or more veins. Every skeletal muscle fiber is supplied with a nerve ending that controls its activity. Unlike cardiac and smooth muscle cells, they do not need a nerve supplementation. Skeletal muscles have a very rich blood supply, okay? It makes sense. You need to move all the time. Skeletal muscles, the main job is movement. So you need a lot of nutrients and a lot of glucose and a lot of blood. Now, according to this, why do you think we need more veins serving each muscle than arteries? Exactly. Because you work more, you excrete more. General rule. So we need more veins to take away those nutrients. What happens if it keeps accumulating and you don't excrete them? What happens to the muscle? Fatigue. Perfect. Okay. Now the connective tissue sheets, okay? For this one, I'm still using the pencil case rule. Basically, let's say I have a bag, okay, that has 10 pencil cases inside it. Each pencil case has 10 pens inside it, okay? This bag that covers the entire 20 pencil cases is the epimysium, okay? Each pencil case represents a fascicle, okay? Between each pencil case and the next, there is also a connective tissue layer called the perimysium. Peri means around. And then each pencil case has inside it many pens. Each pen represents a muscle fiber, exactly. Between each muscle fiber and the next, we have the endomysium. Endo means inside. The pens themselves also have organelles inside them. So the pens are the actual cells of the muscle. The muscle fibers are the cells. Inside those cells, you have organelles. Inside any cell, you have organelles. What are the specific organelles present in the muscle fibers? Myofibers. So myofibers are basically just fancy organelles that are only specifically found in the muscles, muscle fibers, okay? So you need to know that epimysium is dense irregular connective tissue. Perimysium is dense irregular connective tissue as well. And the last one, the endomysium, is irregular connective tissue. What are the functions of the connective tissue sheet? Why do we have the connective tissue sheet? Of course, protection. Aid in movement by pulling on bones. So it's basically saying, when you have this connective tissue layer is continuous with the bone. So if I want to move, the muscle pulls on the bone using this connective tissue layer. And there are specific connective tissues that are going to attach to the bone that we're going to see next. So provide routes for entry and exit for blood vessels and nerve fibers. This is important. So how does the blood vessel find its right way to enter the muscle? It's through these connective tissue sheets, okay? It uses as its own path. Now we have the attachments. What do I mean by attachments? Attachments to what? Or of what? It is the attachment of the muscle to the bone or to any supporting structure. It could be direct or indirect. Okay? What do I mean by direct or indirect? Indirect, I'll go with indirect first because this is the more famous and more common one. Indirect is either a tendon or aponeurosis. Basically, this is the bone, okay? This is a scapular bone and this is the radius bone. This is the biceps, this is the muscle. The muscle attaches here and here using these tendons. So tendons is the specific specialized connective tissue that connects the muscle to the bone. There are two types, okay? There is the origin and the insertion. Basically, origin from its name is where the muscle originally attached and it barely moves, okay? It doesn't move that much. Whereas the insertion is where the muscle inserts into the bone, and this one is the one that causes the movement, okay? Am I going too fast? Too slow? Okay. Now, aponeurosis. How is tendon different than aponeurosis? Basically, it's the same thing, but it's specialized. How? When it goes, it's not a normal tendon, but when it arrives at the palm, it spreads like a sheet. It's more stronger, way stronger, okay? There is also a few more aponeurosis in the head and around the neck. This is indirect attachment. What do I mean by direct attachment? I mean, it looks like the muscle is directly attached to the bone. It's like me saying, I got the bone and I put the muscle above it. This is direct attachment. It looks direct, but it's not actually direct. If you look closely with the microscope, you will see connected collagen fibers, okay, and actual connective tissue fibers that connect the bone to the muscle. But it looks like it's directly attached to the bone. So the indirect is using a tendon and an aponeurosis where I actually have an origin and an insertion. Origin barely moves, it's very important. I think this is what came in the exam last year, to know that the origin barely moves and the attachment, the insertion moves. And the attachment pulls to the origin. The insertion pulls to the origin. Yeah, that's it. Any questions so far?
Okay. Now for the next section, skeletal muscle fibers contain calcium-regulated molecular nucleus. We're going to talk about the functional roles of specialized organelles of the muscle fibers. And for convenience, I'm going to say muscle cells instead of muscle fibers, so you don't mix between the fibers, the fibrous and the fibers. And we will describe the slide now. So now, Mohammed already mentioned the myofibrils, which are specialized organelles for the muscle cells. Other things we can notice about the muscle fibers, or the muscle fibers, which are the muscle cells, is that the nuclei are pushed to the periphery. And why do you think this happens? Why do you think the nucleus or the nuclei are pushed to the periphery or to the plasma membrane, the sarcolemma?
Because in the middle is filled with cytomeres.
Yeah, yeah, because myofibrils constitute around 80% of the cell's volume, and so they push a lot of structures to the periphery of the cell. And we have a huge number of mitochondria to generate energy, and we said that anything related to the muscle, we use the word sarco. So sarcolemma is the plasma membrane, sarcoplasm is the cytoplasm, and sarcoplasmic reticulum is the smooth endoplasmic reticulum. Now, something important you should know that in the muscle cells we have glycosomes and myoglobin. Of course, you know myoglobin, it's similar to hemoglobin, which carries oxygen in the blood, but this one is specially for muscles. And glycosomes store glycogen. Now, when we take in food, we take glucose, huh? We should store glucose, and we store them, we polymerize them, and we store them in the form of glycogen. And whenever we need energy, we break down this glycogen into smaller glucose units, and then we utilize the energy. And this is why we have a huge number of glycosomes in muscle cells. In addition to that, we have the myofibrils, sarcoplasmic reticulum, and T-tubules. We're going to talk about them. Myofibrils are cylindrical bundles. They're made up of the myofilaments. We have actin and myosin mainly, and they are arranged in sarcomeres, which are the contractile units. So when the muscle is going to contract, the changes happen at this level, at the sarcomere. Okay? Let's look at this very closely. This is a sarcomere. Sarcomeres will make up the myofibril. What is a sarcomere made up from? It's basically the arrangement of actin and myosin among each other. So we have areas. The sarcomere starts from one Z line and ends at the other. So this is the Z line and this is one sarcomere. Okay? We will have areas that are dark or that contain myosin mainly, it's the A band. A good way to remember that the A band is dark and contains myosin, the thick filament, is remembering that the word dark has an A, so it's the A band. And the word light has an I, so it's the I band. So this is how we know it's light or dark. And the H zone in the middle does not have any overlapping. It's just the myosin. We don't have any overlapping here. It's hollow or empty. And yeah, so this is the H zone. The M9, the middle, has myosin attached there. Proteins, which is myomycin, which attaches myosin at the center or the M9. And this is basically, yeah, this is a dark zone because it has proteins or accessory proteins attached to myosin. And at the Z line, we have anchoring of actin. Okay? And yeah, as I said, the I band is the light area which has only actin. So this is basically the arrangement. One sarcomere has the A band and half of the I band. Okay? You can remember, you know, we always use the phrase from A to Z, so you can say that actin A is anchored at the Z line, and yeah, from Z to Z is one sarcomere. And Mohammed has a mnemonic. Would you try to explain?
I usually use this one. It's called Zayed is a hungry man. Okay? So Z is the last alphabet. So it represents the end of the sarcomere. And it's attachment of thin filaments. M is middle. And this M line represents the midline of the sarcomere. It's the midline of the sarcomere and the attachment of thick. So the thick attaches at M and the actin attaches at Z. I is a thin letter. I band has only thin filaments. H is thicker, okay? So it has only thick filaments. And if you mix the H and the I, you get an A. So it is the hybrid of I and H, and A band has both thin and thick filaments, remains constant during contraction. This is very important. I can guarantee that 50% chance to get this question tomorrow. Okay? So I, if you look at it, it's actually an H, but it is hybridized with the I. Okay? So it has one. And A does not change. Okay?
Okay. So هلا very quickly, what is light and what is dark? What areas appear light and what areas appear dark? And you guys know that this arrangement of actin and myosin is what causes the striation. صح? Because when you're examining the sarcomere or you're examining the myofibrin, you have arrangement of the sarcomeres, one after another. And we have areas where we have concentrated myosin filaments or overlapping. So it will appear, the areas that are overlapping will appear thicker or striated when you examine it microscopically. Now, what is light and what is dark? The H zone is light because it only contains myosin. We said it has no thin filaments. It's in the middle and it's bisected by the H zone. So yeah, the H zone is light, only myosin, no thin filaments. The I band is also light because it has mainly actin. So you can think of the word high as the light area. And then you have the M line, it's dark because of myomycin, which is connecting myosin and titin. And the A band is dark, we said dark has an A, so it's the A band. And the Z line is also dark because it is anchoring actin. So the areas where we have anchoring of the filaments, M line and A, M line and Z line will appear darker. And the A band is dark because it has overlapping. The H zone is light because it only has myosin, no overlapping. And the I band is light because it only has actin, no overlapping. Okay, now I'm not sure how relevant this is, but I'm explaining it anyway. If we cut through the I band here, in this area, we will see only the thin filaments. We said that right now. H zone, only myosin. The M9, we're going to see accessory proteins and this sort of linking. And the outer edge of the A band, we said we have the overlapping, so you will see the arrangement of myosin and actin along each other. And this is a hexagonal arrangement. So we can see that one myosin is surrounded by 6 actin filaments, and one actin is surrounded by 3 myosin filaments. Okay? So now I want you guys to match for me which one applies for which. Okay. Is it clear that? Yeah. contraction. Yes, it's a sarcomere. Yes, and the lighter central portion of the band, the H. Okay. So the distance between two Z lines, we said this is the length of the sarcomere. The contained only actin filaments is the light zone, the I zone. Both actin and myosin are overlapping, A band, definitely have to memorize this. Tiny contractile unit, we said sarcomere is the functional unit of the muscle set. Actin filaments are anchored to these disc-like membranes. We said actin A to Z. They're anchored at the Z line. And lighter central portion of the A band, it is the H. And if I ask about the M9, it is the region where the myosin is anchored.
Is it dark or light?
Dark. M9 is dark. Okay. Helwa, the thick filaments and the thin filaments were between. Thick filaments is myosin. Myosin is made up of 6 chains. 2 are heavy, are going to give me the chain, and then the rest are for the globular head. Okay, and this head is what's going to bind with actin. And we also have an ATP binding site. So when I bind ATP, the muscle will, myosin is going to detach from actin. And then we use this energy for the power stroke and everything. We'll go back to this later. But yeah, we have to know that the tail that is connected to the M9 does not have a head. So the head is only at one side of the myosin. And the head has an actin binding site where it's going to bind actin, and an ATP binding site where it's going to bind ATP. The thin filament actin, they are actually globular proteins. So it is a protein called G actin or G protein. These proteins are what bind myosin. When I arrange them, they polymerize into longer chains of actin filaments, and we call them filamentous actin. Okay, so filamentous actin is actually made up of the G proteins that are connected to one another. And we have 2 very important regulatory proteins, troponin and tropomyosin. Tropomyosin is what blocks the binding site when I don't need contraction, when I don't need binding. And troponin is what regulates when or not I'm going to uncover my binding site. So, I hope I'm correct. Okay. Tropomyosin blocks myosin binding sites. Troponin is what's going to uncover the binding sites for me. We have 3 very important subunits. One is going to bind troponin to actin, and one is going to bind tropomyosin to actin, and one will receive the calcium ion. So when I receive the calcium ion, I'm going to change my conformation and uncover the binding sites. Okay? The other proteins, elastic filament titin, we said has been recoiled. Dystrophin has the homeostatic imbalance, we're going to talk about it now. Other proteins, forget about it. And descendant filaments, also for connection and something. We have something called muscular dystrophy. It's a disease. Duchenne muscular dystrophy is X-linked. I saw that a lot of you guys are confused with X-linked recessive and all that, so I put a slide here. X-linked means that this disease is carried from a mother, mainly from the mother to the child. And X-linked, now we know that the males have an X and Y chromosome, and females have double X. Now, when I am making a baby will get one X from the mother, and either an X or a Y from the father. If they got a Y, then he's going to be a male, because X and Y. If they got the X, then they're a female. And if this is X-linked recessive, means the defect is on the X gene. So if I will give or pass the defective X gene to my child, and he will definitely get a Y from the father, so he will show the disease. Yeah, he got the X with this type. This one is the defective X gene, right? So this male got it from his mother, and he got the Y from his father, and so he has the disease because he got the defective X. And this is what X-linked means. So this is X-linked means it mainly appears in males, and we have a mutation in the dystrophin gene, and so dystrophin cannot here. It usually stabilizes the sarcolemma. If it's absent, once I have contraction, my sarcolemma or the plasma membrane is going to tear apart, and I will have influx of calcium, which will destroy everything inside. Okay. هلا smooth endoplasmic reticulum which regulates intracellular levels of calcium. This is the sarcolemma, which is my plasma membrane. When I receive a when I receive it, it's going to pass through my plasma membrane, and then it will implement the action on my organ or my cell. فهلا the nerve is going to send the impulse or the action potential to the sarcolemma. And we know that the sarcoplasmic reticulum is the smooth endoplasmic reticulum of muscle cells, which regulates the level of calcium. So, now why do I have invagination of the sarcolemma? Surface area. Whenever you have folds, it's always for increasing the surface area. So now, this invagination of the sarcolemma is what's going to travel, send or propagate my signal faster, and for it to reach for it to cover a lot of my muscle fiber. And so, this invagination will send the action potential right here in this invagination of the sarcolemma, which is the T tubule. And then this, my sarcoplasmic reticulum, is going to have voltage-gated channels, which will sense the change in my voltage and then release calcium. Once the calcium is released, we have the binding and the interaction. So, terminal sisters are the sarcoplasmic reticulum in either side of the T tubule. T tubule is the plasma membrane which invaginates inwards to propagate the action potential faster for the sarcoplasmic reticulum to release calcium. The sliding of filaments. Everyone thinks that when we say contraction, we mean shortening of the muscle, but this is incorrect because the filaments are only sliding over each other, so the overlapping is increasing. That's it. We're not actually getting shorter or anything. At the microscopic level, I don't think I have a picture. I have. What happens here when I'm overlapping, the myosin is going to pull the actin towards the end. It's trying to pull it. So it will be pulling it from either side. Myosin stays the same length, it's not changing at all. You can see the A band remains the same. Okay, but actin is being pulled towards the midline. So it's going to appear as if my eye bands are getting shorter because they are overlapping more. Okay? This overlapping is going to come as close as possible, so I'm going to cover my H zone entirely. The H zone is going to have some degree of overlap. So my H zone will disappear because I have overlapping in that area. My eye zones are going to get closer to one another because they are being pulled towards the center. And so the Z9 are going to get closer because they have actin attached to them. So when actin moves, the Z9s are moving as well. Okay? And then what else I was going to mention? In A band, they did not change length. That's the successive A bands. So this A band and the one closer there are actually getting closer to one another. لأنه my Z9 are getting closer. So the sarcomere is shortening, and so the distance between the two A bands is appearing to be short. Okay. Any questions for that section? Okay. Now moving okay. Action potentials. What do you guys know about action potentials?
They get lots of questions from this section.
Change in the membrane potential.
Yes, the nesting membrane potential is changed. This happens because I have a charge across my plasma membrane, and this charge is basically what allows ions to either enter or leave. Stop because it's a gradient, electrochemical gradient. And so the action potential is when I have changes in my plasma membrane charge due to, sometimes it's due to ions entering or the channels opening, and we have flow of ions in and out and on. So in muscle cells, what you have to know, both muscles and neurons are excitable, which means they react or respond to electrical signals. A chemical messenger or a neurotransmitter is something that's going to be sent or travels to the area I want to perform an action, and then what happens is the neurotransmitter is a chemical. So this chemical will bind to chemical gated ion channels. Yeah. So chemical gated means once I receive a chemical, I will open up and then I will let the ions flow in or out. Okay? And when the ions are flowing in or out, I have a local depolarization or a local change of the charge at that area. So once this happens, voltage-gated channels, which respond to changes in the voltage, will open up as well and then propagate my signal. Okay? So it starts with the chemical binding to the chemical-gated channel. The chemical-gated channel will cause a local depolarization or change in the membrane potential. It's local because this chemical acted on this channel, and this channel is what allows the flow of ions. And then once this happens, the sensors or the voltage-gated channels, which are around or near the chemical-gated channel, are going to open up as well and propagate my signal. Okay. Okay. Guys, Helwa, what we have to know is we have the motor neurons that are going to control or allow me to move and perform actions and everything. Will have axons. You know that a neuron is made up of the cell body, which is going to transmit the signal along the axon to the effector organ, which is a muscle or a gland. If it is a muscle, I'm going to produce movement. If it is a gland, I'm going to secrete hormones. This axon is going to penetrate a muscle, and it will branch there to try and contact as much muscle fibers or muscle cells as it can. Okay? Each muscle cell is only innervated by one axon. Okay. So here we know, let's look here closely. This is the end or the axon terminal, which has the synaptic vesicles, which has the vesicles that contain acetylcholine, the neurotransmitter working in it. So this one is actually not in close contact with my plasma membrane. We have a space called the synaptic cleft. And in this cleft, we have the release of acetylcholine, which is going to bind to the chemical gated channels on my muscle, and then we're going to form the local depolarization. Okay? Now, how this happens is that we have, once the axon terminal receives the signal, calcium is going to be released, and then acetylcholine releases as a response. Acetylcholine will bind to the chemical-gated channels, and then the chemical-gated channels will cause depolarization. This depolarization will propagate to voltage-gated channels, which are going to transmit my signal all over the cell. And then, now we said the local depolarization is called the end plate potential. Okay, that is caused by acetylcholine binding that chemical-gated channel. It triggers an action potential in the adjacent plasma membrane, and then the action potential travels down the T-tubules. We said that the T-tubules are invaginations of the plasma membrane. So this action potential is going to travel inside, and then causes the sarcoplasmic reticulum to release the calcium. Now we have the calcium released into the cytoplasm. What's going to happen? Calcium will bind troponin. It's going to bind to troponin, and then once it binds, troponin will cause a movement of the tropomyosin to expose my binding sites. Once the binding sites are exposed, myosin will bind to actin and form the thrust bridges. And then we have the power stroke, the movement, and all of that. Okay. This is basically what I just explained, the end plate potential and how you propagate your signal. And yeah. And normally our plasma membrane or the fluorescent membrane potential, it is negative on the inside and positive on the outside. So whenever I have an influx of sodium ions inside the cell is going to become more positive. And this is what we mean by depolarization. Which of these events must occur first to trigger the skeletal muscles to generate an action potential? It is true. Acetylcholine shows the temporary permeability to sodium. We said when acetylcholine channels, it's going to open up, so sodium is going to enter the cell temporarily, just until this is over. But then, by then, we have depolarization, and my voltage gated channels already sense this change in the voltage, and then they propagate my signal. So it's okay. Which one of the following functions do calcium ions perform during skeletal muscle contraction?
Two.
اللي هي expose myosin binding sites on the actin, which when they bind troponin.
For example, if it was actin tendency instead of myosin, we can choose 4, right?
هلا calcium actin yes. لأنه myosin ATP is what binds there for power and energy.
I didn't understand.
Hi. She said if it was actin.
Yeah.
So when you bind calcium, what it means is if this was actin, means calcium is binding to actin for them to change shape and cause a contraction. If this was actin, then it would be there. But it's myosin, which is incorrect, and what binds myosin? ATP. And هي troponin on actin. Troponin is the regulatory protein on actin. So once calcium binds troponin, again troponin has 3 binding sites, صح? One for calcium, one to attach it to the actin, and one to attach tropomyosin to actin.
So when calcium attaches to troponin, we're kind of attaching the actin.
Yeah. But basically, you just know in the بالضبط on troponin to expose the binding. And by changing the troponin. Okay. Now here, the cross switching. First of all, after contraction, what happens is now I have a large number of calcium ions in my cytoplasm, because of contraction. The sarcoplasmic reticulum wants to claim this back. So we have pumping and active transport for the calcium to get back into the sarcoplasmic reticulum. And this is important because we don't want contraction all the time. Because as long as we have calcium in the cytoplasm, we have cross-bridging and all this. And so we have to reclaim our calcium back to prevent contraction. This part that you just mentioned, this one? We said that when we receive an action potential that travels down the tubules, the sarcoplasmic reticulum will release calcium. So once calcium is released, the calcium will bind to troponin. And then what happens is that troponin will uncover the binding sites. It's going to move tropomyosin out of the way. And when this happens, myosin will immediately bind to my actin, and we will have the sliding and the overlapping. So if I have calcium all the time in my cytoplasm, I will always have contraction and binding and all this, and I don't want that. So the sarcoplasmic reticulum is going to reclaim the calcium back and store it. Okay, it's an active transport process in which the calcium is gained back in the sarcoplasmic reticulum to prevent overcontraction and exhaustion. And now we have a condition called rigor mortis. Okay, let me explain this one first, and then we can go to rigor mortis. In the cross-bridging, what happens is the myosin already has ADP and phosphate from the previous contraction. It already has this. And now it binds to my actin, and then it will release this energy out. It's going to release my ADP and the phosphate away. This is the power stroke, the energy it received from ATP. Now I release the ADP and the phosphate, but I still have binding. So what happens is an ATP is going to come in, bind to myosin again to detach it from actin. So yeah. Now when I attach ATP, bind ATP to myosin, I'm detaching it from actin and giving it energy enough to go on and do this again. So now ATP is hydrolyzed or broken down again into ATP and phosphate. And now here it has energy and it is in the high energy state. And again, it's going to bind to my actin, release the ATP and phosphate, and then by releasing them, it goes in and it does the power stroke thing, and it moves and pulls the actin, and then another ATP will connect, I will detach, and then use this energy to bind again and power stroke. Okay? So why is ATP very important in this process? Detachment, exactly. Helwa, rigor mortis is something that happens after death. what happens after that is that the calcium remains in my cytoplasm. I cannot replace that with the sarcoplasmic reticulum. So when we have calcium all the time, we have attachment of myosin and actin. Because they are dead, they are not producing ATP, so they cannot detach actin from myosin, and we have a constant state of contraction. Like, is it clear?
Yes.
I have this video. It's actually this was a PDF. Now I cannot open it. It showed how this happens exactly. It's very explanatory. Okay, the following is a list of events that occur during a muscle contraction. What is the correct sequence of these events? طيب. Would you mind explaining ولا؟ لا هي دي صح. It is D. Okay. أول شيء في calcium ion. Calcium ion is released from the sarcoplasmic reticulum. And then 5, calcium ion bonds to glutamate. So we know that these are very important and they are the first steps in contraction. فأكيد you're going to exclude options A and B. هلا 1, myosin cross bridges bind to actin. Very clear. This is the next step because now I uncovered the site. So 2 or 4, what happens next? 2, the free myosin head splits ATP. And 4, the myosin head pivots toward the center of the sarcomere. Why is this one the correct option? Because we said that here, the myosin already is in a state of readiness to go on and bind and move forward. Because it already has ADP and phosphate. Once it moves forward, I will release these. And then another ATP will bind for me to detach and use this energy for the power stroke again. So this is why the myosin head pivots toward the center of the sarcomere is the correct answer. And then the myosin head binds an ATP molecule to detach. The free myosin head splits ATP. So once I detach, I will split it again into ADP and phosphate, and I will repeat this one later. Any questions?
Okay, so what are the properties of whole muscle contraction? Differences between the muscle's tension and load is the first objective in this section. What is the difference between the muscle tension and the load? Muscle tension is the force exerted by the muscle on an object. Okay? So this is endogenous. So this is what I excrete. But the load is what I receive. So every action has a reaction. This is the reaction. So the load is what I'm carrying. So if I'm trying to carry something like my bag, what I'm doing is my muscle tension, and the bag, the heaviness of the bag is the load. Okay? The motor unit. This is essential. This is the concept comes in many questions. It's a high-end concept. What is the motor unit? The motor unit consists of the neuron and all the muscle fibers it innervates. Every motor neuron and the muscle fibers constitute one motor unit. Okay? This is important for the concept of the frequency and the strength we're going to do now. So, the muscle twitch, this is what scientists use to study the muscles in the labs using a myogram. It is the response of a muscle to a single stimulation. What happens? This is what you get from a myogram. This is the curve. You need to know that this curve is specific for each muscle. Each muscle has a specific curve that is different in a few ways. We'll explain that. Every curve has a latent period. Okay? This one. The very first one. This very small line. What is the latent period? It is where excitation contraction company is happening, but there is no muscle contraction that can be detected by the device. So they are starting to initiate a contraction, but no contraction is still detected from the device. Now the period of contraction, this is where the peak happens, peak of tension development. So the cross bridges are active, and I get a maximum peak. Okay, and then the period of relaxation is where the calcium ions are pumped back to the sarcoplasmic reticulum and declining active cross bridges. Again, this is specific for each muscle. Some muscles will have a longer latent period, but still longer contraction, period of contraction, but it's not that high as tension. So a muscle can provide very strong tension at a very short time, or little bit of tension, but over a longer time. Okay? This is the important part. The graded muscle contraction. What do I mean by gradient? I mean I have more than one stimuli that are stimulating the same muscle. The first 2 points are the key points here. When I increase the frequency of stimulation, okay, this causes temporal summation. What do I mean by increasing the frequency of stimulation? It means for the same muscle, I'm providing more stimulus. So how many stimulus I provide per minute or per second. Okay? This causes summation, temporal summation. This one. So the muscle tension happens, okay? بعدين the relaxation happens, but before I reach maximum relaxation, there is a second stimuli that will synergistically add on the previous tension, the previous tension. So I get a higher strength of contraction. So the higher the frequency, the greater the strength of the contraction of one specific motor unit. So I'm activating just one motor unit, but multiple times. The second one, I'm increasing the strength of the stimulus, where I activate more motor units, but not high frequency. So the stronger the stimuli, so if the stimulus itself is much stronger, I'm activating or recruiting more motor units. This is called recruitment. The stronger the stimulation, the more motor units are activated and the stronger the contraction. Okay? So, if I have one motor unit and I give it too frequent stimulus, I get temporal summation. And if I keep increasing this frequency too much to the point where relaxation is barely happening, I will reach a maximum stage where I can't add more tension. Okay? So, you need to understand the difference between incomplete or unfused tetanus and fused or complete tetanus. What happens is, in unfused tetanus, you are giving the maximum possible amount of stimulus that the muscle can take at a specific time. I keep adding more and more stimulus, and I can barely see any relaxation. However, every muscle has a limit. I can't just keep adding stimulus forever. It will reach a limit where there is barely any relaxation happening, to the point where you will find that the person is contracting, just contraction. There is no, the calcium ions don't leave the binding cells. There is always contraction. In real life, this never happens. The body will never allow fused tetanus to happen, because if fused tetanus happens, death, rigor mortis. Think about it. If I have just contraction, no relaxation, خلاص. A very important point to note: in unfused tetanus or just fused tetanus, what is being respected in the terms of intentions? What is something I cannot just escape? Repolarization. The hyperpolarization people. No matter how small the stimulus is, if the muscle has not undergone the repolarization, I will never have the contraction. I have to wait until repolarization happens. Okay? These come in almost every exam. Every class paper will have a question about the fused unfused tetanus and the temporal stimulations. Okay? If the muscle is stimulated increases the faster rate, the relaxation time between teachers becomes shorter and shorter, the concentration of the cytosol rises higher and higher, and the degree of wave summation becomes greater and greater.
Guys, I have a question. What is the period in which I can no longer react to the stimulation I'm receiving? Why does refractory period happen?
Which of the following is answer this question? Now, which of the following is most likely result if one nerve stimulus arrives at the muscle fiber so soon that the fiber is only partially relaxed from the previous twitch, and why? Why?
Because it was partially relaxed. If it was not relaxed, then it would be completely relaxed.
Exactly. So you need to focus on those things. Okay? Now the stimulus strength. Now I spoke about how the muscle responds if I increase the frequency. I get summation. Now if I increase the strength, what happens? I recruit more motor units. Basically as if I'm shining dim light to your eyes, only one motor unit is activated. However, if I shine a very much stronger light, so many more motor units will be activated. So the stimuli that produce no observable contractions are sub-threshold stimuli. There is a threshold stimulus that after this threshold, I can actually see a contraction. Anything below the stimulus, I will not have any response. The stimulus at which the first observed contraction occurs is called the threshold. Beyond this point, the muscle contracts more vigorously as the stimulus strength increases. So, from then on, the stronger the stimulus, the higher the response. Until, of course, again, every muscle has a limit. Until I reach maximum stimulus, after which the contractile force will not change, no matter how much stronger I provide the stimulus. Now, this is important, the size principle. So now I said, the stronger the stimulus, the more motor units I'm going to recruit. However, does this happen randomly? Like, do I just randomly pick a motor unit to activate? No. There's a size principle. What the size principle tells us is, the smaller motor units will be activated first. Why? Because they're easier. They are more sensitive to the changes in the ions. However, the bigger motor units, the ones that innervate way more muscle fibers, they need a stronger stimulus to actually be activated.
Least excited.
Sorry?
Least excited neuron.
Yes, exactly. So the bigger, the least excited. Motor units with the smallest muscle fibers activated first because they're controlled by the smallest, most highly excited motor neurons. Now, as you go larger and larger, the contractile strength increases and the bigger motor units are starting to be excited. The largest motor units containing large coarse muscle fibers are controlled by the largest, least excited, highest threshold neurons and are activated only when the most powerful contraction is necessary. Now, the concept of muscle tone, it came from us last year as well. So it is the constant, involuntary contraction of all muscles that stabilizes joints and maintains posture. And it happens due to spinal reflexes. Without this, what happens? Without the motor tone? Death. Exactly. So this is what maintains our posture. Also, a very important point to note in muscles without constant innervation and stimulation will definitely atrophy, will lose its mass. Muscles can never ever survive. It can lose 5% of its mass every day if it's not innervated. So it is essential to have this muscle tone. Now the isotonic and isometric contractions, it's an easy concept. Basically, how I memorize this, isotonic contraction is any contraction that causes a change in the muscle, the length of the muscle. Okay? So either it becomes shorter or longer. Isometric is where I don't have a change. Iso means same. So how I understood this was isometric means same meters. So if this was 0.5 meters, it stays 0.5 meters. So it doesn't change. There are two types of isotonic contraction. There is the concentric and the eccentric. Concentric is the normal one we always see. So this is concentric, and is when it becomes shorter. However, some muscles, extending its length, provides force. This is eccentric. Eccentric is away from normal. Okay? So normally you have the muscles shortening. So abnormally you have muscles lengthening. So muscles that provide force when they are stretched, they are undergoing eccentric contractions. Okay? Can anyone give me an example of eccentric contractions? Exactly, 5-1. Also, when I put something down, the triceps are extending and they're using force. I don't use my biceps to put something down. I'm majorly using my triceps. Okay. Yes. Almost.
Now, ATP for muscle contraction is produced aerobically or anaerobically. This is how we are providing energy for our muscle cells to complete contraction. So, ATP supplies the energy to move and detach cross bridges. It's very important, not only for this, and the sodium potassium pump, maintaining the plasma membrane retention and everything. So it's very important. So as I'm using ATP and I'm hydrolyzing it into ATP and phosphate, I need to regenerate it as fast as I can, because if I don't have ATP, what's going to happen? Homeostatic imbalance. So this is why we have to generate it as fast as we can, and we have three major pathways. The easiest in here, direct phosphorylation. I have creatine phosphate, and I have ADP. Creatine phosphate is a compound present in the muscle cells 2 to 3 times higher in higher amounts than ATP. So what's happened is this creatine phosphate is the Because you know ATP is ADP plus phosphate. Where do we get this phosphate from? Creatine phosphate. The maximum muscle power for about 15 minutes is enough to energize a 100-meter dash. It is very quickly, but the yield is not that high because 1 ATP per creatine phosphate. And creatine phosphate is actually, you don't want to have like, very huge amounts of creatine. And so this is important for activities that are very fast and very short. Now, anaerobic pathway and aerobic pathway, I will discuss it in this slide. هلا حكينا when we eat food we get glucose. Glucose is the main source of energy in our body, صح? And now glucose is going to perform glycolysis to give me 2 ATP and pyruvate molecules. Pyruvate is a substance that's produced with glycolysis. So glucose is going to metabolize or perform glycolysis to give me 2 ATP and pyruvate. If I have oxygen, I'm going to go to the citric acid cycle and then electron transport chain and produce more 36 ATP. If I don't have oxygen, there will be fermentation and you don't have to worry about this, but if I don't have oxygen, I'm going to have lactic acid. So pyruvate will be converted to lactic acid in the absence of oxygen, and it's going to be converted to 36 ATP molecules if I have oxygen. So here basically, in the liver, lactic acid can be used as a source of energy to give us pyruvic acid and glucose, and then we're going to use this glucose to make ATP. So the heat, this is very fast. We don't have oxygen, but it's very fast. We have 2 ATP per glucose, and in 30 to 40 seconds. If I have oxygen, on the other hand, it takes a lot more time. Thus, we have 36 ATP molecules per glucose. So this one is going to give me a huge or a high yield of ATP, while this one does not. But this is faster. And so I'm going to use this when I don't have oxygen and I need more ATP. And the aerobic pathway is very important for aerobic endurance. It is how long you can go on. I think I have it here. How long you can go on utilizing oxygen to gain energy. And then the moment I have to switch to anaerobic pathway is when I cannot use oxygen anymore, is the anaerobic threshold. And so here you see that ATP is stored in muscle, it is used first. But then now I don't have ATP, so I'm going to do creatine phosphate. Is going to donate the phosphate to ADP and give me ATP. And then if I don't have oxygen, the anaerobic pathway, anaerobic means no oxygen. And so glycogen that is stored will be broken down to glucose. We said glycogen is a polymer of glucose. So I will break it down to glucose and utilize this energy to give me ATP, 2 ATP per glucose. If I have oxygen, and prolonged duration exercises are usually a lot calmer, and so we have more oxygen, and you're going to use the mitochondria to give us more ATP, 36 ATP. And the equation, this one happens in the mitochondria. You're not only using glucose, you use fatty acids, you use amino acids, and all that. Okay. Intense exercise of short duration produces fatigue much more rapidly, but you recover faster. Whereas the slow developing fatigue, which happens for long periods of time, would need more time to heal. Now, what causes fatigue? We have many factors that will cause fatigue. Now, why does muscle fatigue happen? It happens for a reason. Lactic acid causes soreness. So when you play for long periods of time, you have buildup of lactic acid, so it hits your muscles are very sore.
When you have enough ATP and you want to preserve it.
Yeah. We said in the depletion of ATP, rigor mortis happens. I cannot detach myosin, I have constant contraction and all that. So muscle fatigue is like, you know when there is a marathon runner and they're running and running and running, and then like they just have a little to reach the end line, but they cannot do it anymore, so they cannot contract, they cannot run. This is the same thing. So muscle fatigue happens to preserve the left amount of ATP, because if I don't have ATP, completely use all of my ATP, rigor mortis happens. And so this happens due to a lot of reasons. Potassium is lost from the cells. حكينا احنا when we have contraction, the excitation contraction coupling. What happens is we will have depolarization in the membrane, potassium is going to leave the cell and sodium will enter the cell. When this happens, my membrane potential is not very stable. Well, I have the charges are different inside and outside. And so I will not be reacting the same way if I have a normal state. And this is why when potassium is lost from the cell, I have overstimulation, which means that I'm always having an imbalance at the level of the membrane. Increased inorganic phosphate from creatine phosphate and ATP breakdown may interfere with the release of calcium. We know that this has an effect, phosphate and all of this, because we said ATP controls a lot of processes in the cell. Increased magnesium allows voltage-sensitive proteins to decrease the release of calcium. This happens because magnesium and ATP bind together. So if I don't have ATP, magnesium is going to be overloaded in my cell, and this causes ionic imbalances, and we don't want that. Now the excess post-oxygen consumption. Post-oxygen? Post-exercise. Okay. Increased rate of oxygen intake following physical activity. This happens because I want to replace everything that I had lost in my exercise. You want to return everything back to its baseline. You will restore your oxygen reserves by breathing and all this myoglobin, which stores oxygen in muscle cells. The accumulated lactic acid will be reconverted to pyruvate, and then we said that pyruvate, once we need the energy, will be converted in all this. Glycogen stores must be replaced from glucose. Glucose will form polymers and store it as glycogen to use it once we need contraction. And finally, ATP and creatine phosphate must be preserved. And the creatine phosphate should always be there so that whenever I have low ATP levels, I'm going to directly give the phosphate to ATP. Okay, which is a reason for muscle fatigue? A reason. Three. So imbalances in magnesium levels interfere with calcium release. The inability to generate sufficient quantities of ATP due to feedback regulation of synthesis, incorrect. ATP levels enough to cause contraction, this is not muscle fatigue. Inadequate numbers of mitochondria has nothing to do with it.
So now,
Guys, you can see without doing
when we have contraction, of course, the force of the contraction, the velocity and duration is not the same. Not every contraction you make has the exact same force, has the exact same speed, okay, and duration. What determines what? Force of muscle contraction, we spoke about two factors: the frequency and the strength. So when I say force of muscle contraction, I'm talking about the number of myosin cross bridges that are attached to actin. This is the key principle. So the more cross bridges I have, the stronger the force. So we said the frequency increases. We said the stronger stimulus will recruit more motor fluid and therefore cause more contraction. Two extra ones are the size of muscle fibers and the degree of muscle stretch. The size, obviously, the reason why many people go to the gym to build their muscles is because by resistance exercises, they are getting bigger muscles, okay? So they can lift more, meaning there is stronger contractions, okay? What happens simply, I'm getting bigger muscles, so I have more mitochondria, more blood supply, I have more oxygen, more nutrients to supply each muscle fiber, so I'm getting better contractions and more contractions. Again, you need to understand that muscles do not increase in cell number, they increase in cell size, okay? They cannot divide. However, some muscle cells can split because of how much it increases in size, it can split into two, but the concept is muscles can never divide, okay? They only increase in size, it's hypertrophy. Degree of muscle stretch, what does that mean? If you look at this graph, we realize that normally the optimal sarcomere operating length is 80% to 120% of resting length. What does that mean? It means if, hypothetically speaking, the actin and myosin were sat this way, the actin is far away from the myosin. Can you actually have a contraction here? Can you actually stimulate the muscles to shorten and contract? No, because the actin and myosin are not exposed to each other. Same thing here. Here, the sliding is, the distance is so narrow to the point where you can't have us sliding. The whole contraction is basically sliding. So, in this one, I can slide over the whole length of the sarcomere. So the more sliding that is available to me as a muscle, okay, the stronger the contraction. If the muscles are too short, if the actin and myosin are too close to each other, there is no space for me to actually contract and cause the sarcomeres to come together. They're already closed. So I will have very little contraction. Same thing if they are excessively stretched. If you excessively stretch out the muscle, it doesn't actually work. خلاص. It reaches a limit where it will not work anymore because actin and myosin are not even exposed to each other, so I can't have any sliding. Okay? Any questions? Okay. Now the velocity and duration of contraction. Okay? This is influenced by what? The muscle fiber type, load, and recruitment. What do I mean by muscle fiber type? They split the muscle fibers into 3 different categories. Okay? Slow oxidative, fast oxidative, and fast glycolytic. Slow, what do I mean by slow? It means the contraction happens slowly. The movement of the calcium and the binding of the cross bridges happens slowly. Fast means it happens quickly. Oxidative and glycolytic. As my colleague mentioned, oxidative using the aerobic pathways. Glycolytic uses the glycolysis pathways. This table is important to understand, and it is very easy. You don't have to go memorize it word by word, okay? Try to understand it. First, look at this one. Slow oxidative fibers are mainly present or used in exercises that need endurance type activities. So, if you are doing endurance activities, okay, aerobic activities, what are you looking for? You're looking for more oxygen, صح? So, we can say that the myoglobin content is high. You're using more oxygen, so you're using more aerobic pathways. Do you need glycogen to undergo glycolysis and break down to lactic acid? So it's slow. Myosin ATPase activity is slow, of course, because endurance goes for longer periods of time. So endurance activities usually take time, so an hour, a marathon running, for example. Speed of contraction is slow, this is the whole category. Recruitment order, this is the first one, okay? This is the first to be recruited. So, while I'm doing an exercise, the body decides, okay, does he need me to use glycolysis? No. So the first thing I will recruit is this. If it increases in intensity, then I will start recruiting the other ones. So it's always the slow one first, and then the 2 fast ones. The rate of fatigue is slow, obviously, fatigue resistant. Why? I have lower lactic acid produced, and I have lower ATPs produced because I'm using more oxygen, aerobic pathways. So this again, endurance. It's very important to remember that this is for endurance activities. Structural characteristics, they're smaller. There's a lot of mitochondria. Why? Aerobic again. Capillaries, many. Why? Exactly, oxygen. And the color is red because of myoglobin. Exactly. So myoglobin gives it its characteristic red color. And what does myoglobin do? Storage of oxygen. Now fast oxidative and fast glycolytic. So now the body is slow, and then it realizes actually I need to work harder. The slow and fast are the 2 extremes. This one is intermediate in everything. Okay? So whenever you see something that's intermediate, remember it's fast oxidative. So let's go to the other extreme, fast glycolytic. Where is it used? For short-term intense or powerful movements. Example, hitting a baseball or sprinting, high intensity interval training. What do I use? I use fast fibers because I need to act quickly, so the body doesn't just faint. I need the muscles very quickly, so I need to act very quickly. It uses anaerobic glycolysis, as was mentioned. It's faster. Okay. Again, there is very low myoglobin content. I don't have oxygen, and I don't need oxygen because I'm using glycolysis. Glycolysis doesn't require oxygen. The glycogen stores is very high. This is characteristic because if I don't have enough glycogen, I cannot use it in the first place. Okay, and then I have recruitment order is the last one. This is very fast, fatigable. So this one fatigues the fastest, but again recovers the quickest. Short-term intense or powerful movements. The fiber diameter is intermediate. This is the only exception. So the small, largely intermediate, not intermediate is the middle. It has very few mitochondria, very few capillaries, and it is white in color. This one is intermediate in everything. Okay? Intermediate glycogen stores. Intermediate color. Even the color is intermediate. So it's not red, it's not white, it's red to pink. Okay? Then we have lower recruitment. So we said that the muscle fiber type influences the velocity and duration. What else? Of course, the load. The heavier the object you are carrying, the more time it takes. Simple. the fastest contraction is the one where you're not carrying anything. So let's say you're carrying a 50 kg dumbbell, it's going to be very harder to carry and slower, the velocity contraction. Same with recruitment. The more motor units I'm using, the faster the contraction, obviously. Because now I have more people working at the same project. So I have more hands, so I can finish the project faster. If I'm working alone, I will take way more time. If I'm working with a group, I'll finish quickly. Okay? Yes.
So now when we sprint, which type?
The fast glycolytic fibers.
Yeah,
Yeah, it was in the test banks, they said sprinting and fast glycolytic fibers, and I'll come to it here as well. This one, we're talking about the aerobic exercises. It switches to fast glycolytic. From fast glycolytic, switches to slow glycolytic.
So
Both. Yeah. Well, again, you're recruiting more muscles when you increase the intensity of the exercise. So you're going to have different types used. Now, the last section, how does the skeletal muscle respond to exercise? There are two different types of exercises: aerobic and resistance. Resistance is the gym exercises, and aerobic is the endurance exercises. The important one is this. What you need to know mainly is that the number of capillaries will increase when I'm doing aerobic exercises. The number of mitochondria within the muscle fibers will also increase, and they will synthesize more myoglobin. So if I'm synthesizing more myoglobin, what am I doing? What am I changing? If I'm synthesizing more myoglobin, what am I changing? Yeah. In terms of the muscle fiber types.
Changing it from anaerobic to aerobic.
Exactly. So now I'm changing it from glycolytic to aerobic. Okay? Resistance exercise leads to hypertrophy. So, high intensity exercise, the additional muscle bulk largely reflects the increased size of individual muscle fibers, the fast glycolytic variety, rather than increased number of muscle fibers. Okay? It converts fast oxidative to fast glycolytic, but you need to remember, and if you discontinue the routine, it reverts back. It's not permanent. Okay? That's why people say you lose your gains. Yeah, basically it's called metaplasia, you're taking your own. So if you revert back. That's it. The last part is the last genetic imbalance in your section. As I mentioned already, disuse atrophy. Muscles that are not stimulated will diminish in size and will not be able to work anymore. So to remain healthy, muscles must be active. Okay? Under conditions where I have no stimulation, disuse atrophy, I can lose 5% per day of the muscle mass. Even at rest, muscles continue to receive weak stimuli, as we said, the muscle tone. When totally deprived of neural stimulation, a paralyzed muscle may atrophy to 1/4 of its tissue size. Fibrous connective tissue replaces the lost muscle. This is important. So, what happens after I lose the muscle? It is replaced by fibrous tissue. Fibrous tissue is non-contractile. It has no actin, no nothing. Okay? Complete recovery usually takes longer than the clinical normalization. It loses the mass very quickly, but to recover it takes way longer time. This is.
It's not very muscular.
Yeah.
How are we going to share this?
Yeah, I know. So I have to do it. We prepared a Kahoot thing.
We
Sorry,
can
guys.
answer the questions and you guys answer.
Yeah, let's.
Because we wanted to share this screen for you guys to see the answers.
Okay, can I answer questions? Just take questions.
Okay, would you guys want me to read the question out loud and you choose the answers from your phones?
Okay, just join the room.
Okay, open Kahoot and I'll give you the list of
Yeah,
it.
Is there anyone have any question?
I forgot to talk about my study agreed list. Does anyone have a question about it? My study agreed list. And who is that? Yes. I don't. guys know that acetylcholine is the neurotransmitter which eventually causes contraction, right? In my synaptic cleft, I have another enzyme called acetylcholine esterase, and this enzyme breaks down acetylcholine into acetic acid and choline. Okay, and then we take these compounds and regenerate acetylcholine and so on. And so I have receptors on my muscle for acetylcholine. These receptors will bind acetylcholine and cause my contraction. In a disease called myasthenia gravis, in this condition, I do not have these receptors. So acetylcholine is released and then it is inhibited by acetylcholine esterase. I don't have contraction. Acetylcholine causes contraction. So if I don't have receptors for this neurotransmitter, I will not have contraction. Okay?
Yes.
Actually, I have so many questions. It says, which statement best describes the goal of the medication for this disease? Using treatment answer. To increase the number of acetylcholine receptors or to inhibit the function of choline esterase?
Inhibit the function of acetylcholine esterase. لأنه if you have downregulation of receptors, هلا in myasthenia gravis you have this acetylcholine receptors, but their number is much less because if you don't have them, you will have contraction at all. So you have them, but the number is decreased. You cannot actually enhance or increase or upregulate the number of receptors. So what happens is I will be blocking acetylcholine esterase so that acetylcholine, as much as I can, is going to bind these receptors. And you want the binding to happen, but what are you going to do? Remove the inhibition. Okay? Any other questions?
Wouldn't there be any side effects
For
inhibiting acetylcholine esterase?
not really, because you need the binding of acetylcholine to the receptors.
Whereas also we have continuous stimulation.
And
Don't we have continuous stimulation like
but
this?
you don't have the
Yeah,
receptor.
you're breaking the receptors anyway, you know?
Because without the receptor is no contraction. But you're not going to get into side reaction, يعني side effects. Let's just know the basic principles.
Yes.
Increase the number of neurons.
Yes, motor units. Yeah, basically more neurons are stimulating more neurons. The other one, the same muscle is being stimulated again and again.
The cross-bridge cycle. The first picture we saw that it's already attached to an ADP and ATP. So in the normal state, without any contraction, is the myosin attached to anything? So how does it begin? The first.
Before the attachment, شوف هاي this step. Here we don't have any attachment, صح? We don't have attachment, صح?
Okay, so we have the ATP.
هلا this ADP and phosphate are there from my previous contraction. You have ATP and phosphate attached to my
Without any previous
You
contraction.
can't, because we have muscle. This is the thing. If you stop the stimulation of the muscle, it atrophies. There is always very minimal contraction. You cannot escape.
The first contraction in your life.
The first contraction in your life,
هلا you have ADP and phosphate binding to myosin at all times. This هلا when I have the binding, this will be released for me to do the power stroke, and then this will keep contracting until I attach ATP to detach it. The ATP once it binds to myosin, what happens? It breaks down into ADP and phosphate. Fine? And this is used for my second cross-bridge formation.
Okay, so now in the relaxed state, we should see an ATP attached. In the relaxed state.
United States.
What do you mean, this one?
No, like
This-
now, if I عملت هيك زي شو مصر تبعك
Yeah.
وانا relaxed,
Yeah.
okay, مسكت بكتشف انه ATP ولا ATP.
Has to be ATP, yeah, relax. Because if I'm
ATP is detachment. هلا
Okay, إذا relaxed شو هو البيكتشر لازم اشوفه إذا أنا relaxed. إذا أنا relaxed بدي أعمل contract.
relax means no cross-bridge formation, which means it's probably one of these. ATP يعطيك energy, so when you bind ATP, you're detaching, but it is in a state ready to pivot once it binds active.
طيب ماشي أعطيك بدي attach.
Okay.
But how does the last contraction end? هذا السؤال.
You can't end the contractions forever.
It's detaching ATP.
Yeah,
It's detaching.
Yeah, I relax. Okay, خلاص. I remove the ATP. Sorry, I remove the ADP and PI and I'm attaching an ATP, صح? I'm now ready for the next contraction.
حلو. Okay. So now with relaxation, for the next contraction.
This nanosecond where there is no cross bridging and there is an ATP, This is relaxation.
But there is some seconds where the muscle is
It is.
complete relaxation.
Yes, it may seem to be completely relaxed. Again, we have muscle tone.
The way
It is constant and involuntary.
not the muscle, I mean like one myofibria in the muscle, it will be relaxed, right?
It has to be that.
For 1 second, okay. So in this second, where it is relaxed, ATP.
80.
80.
Because as long as, so ADP and PI are only there when I have a cross picture. Okay?
ATP and P.
Yeah, so ATP when I have no cross picture. And I got your question, you know, how did the first construction start? What was binding to the myosin? Yeah, it's ATP. So as long as there is no cross picture, it's ATP.
It's ATP.
Yeah. Now I'm ready to bind to the next one.
Okay, so when calcium attaches to the receptor, the troponin, right?
Yes.
The tropomyosin,
Yes.
okay, and we have available binding sites active. So now the ATP will be converted to ADP and P in order to do the
Cross bridge.
attaching
Yes.
And after attaching, they will be removed to push.
Yes. So stroking and then release of the ATP and PI. And then getting an ATP attached.
So the hydrolysis of ATP to ATP and ATP happens here. This is the first thing that happens.
Yeah, but again,
muscle
it's a cycle.
relaxation مش بس ATP
Cycle.
and ATP. You have the calcium that will go back to the sarcoplasmic reticulum. Tropomyosin will cover the binding sites. Will happen. ATP is always ready in a high energy state. لأنه the moment it binds active, I will do the power stroke and sliding of the filament. So I need to detach and then, يعني, you imagine it as this is that myosin and active, and then they're sliding over each other. So you always need it to detach and attach, detach and attach. To end the contraction, you will need to remove the calcium, you need to cover the binding site, detach and all this. When I detach, ATP is binding. So ATP رح يعمل لي إياها in high energy state. And you know that it is an ATP. So it will break it down to ATP and phosphate. Myosin is an ATP, which means it breaks down ATP, hydrolyzes it. So it's gonna give me ADP and phosphate. But here it's in high energy state. Once I bind actin, I will move forward and release these compounds.
No, here this is a high energy state. No, this is a low energy state.
No energy.
For the ATP one?
Yes.
Because I didn't bring the ATP yet and get the energy from the phosphate.
Okay.
From the phosphate.
So when it's relaxed, when
ATP is one.
ATP, low energy.
Low energy.
And then when the sites are active for the attaching of the heads, ATP is
Hydrolyzed.
hydrolyzed to ATP and B in order to attach and push. And ATP and P are important for the attachment or the pushing, or both.
هلا هم they are ATP gives you the energy. This is the energy. Once I move forward, I will be releasing them. This is how you use the energy.
So just pushing.
Yeah.
The push, the
Basically,
mechanical
the power
push.
You use the energy for pushing, not for attaching.
Yeah, the attaching is done by the fact
Attaching
that
is when you uncover the
yeah, the
binding
uncovered
site.
side. يعني when it's exposed, it attaches. But to move, I hydrolyze the energy. The mechanical action, basically.
لخبطتك؟ خلصت قلت لي بعت لك اياها So does anyone have a question about anything?
Any specific
Anything we mentioned, we didn't mention?
That's fine.
The question you asked, the first question you asked. What after the
طيب بس سو اسمع خاصة
This is the code.
I need to answer this. I'll tell you the question.
guys.
Guys, again, I want to tell you that the quiz will be easy. If you guys understand the basic concepts, the main ideas, you can do it easily. And I don't really remember much from our quiz, but Zakar, they gave us all 5. Zakar?
They give you all 5?
All 5.
Why?
There were problems بالlockdown browser,
بعدين they fixed,
بالconnection,
they added an extra quiz.
So everyone got a 5 بالمصير. Inshallah, انت حتكون زينا. But it's very easy. It was one of the easiest actually.
Anyone wants to answer comment again? خلاص؟ Okay.
احنا we're going to read the questions for you لأنه we cannot display them. It happened.